Hello, everyone. I'm Bang-Lin Song, professor at Yonsei University Medical School. Thank you for participating in the COVID-19 session of the second Korea-Sweden R&D Network Day. I would like to use this privileged opportunity as a moderator to invite 6 eminent scientists from both countries today and to discuss and share our recent research findings on the new coronavirus. So let's start with the first presentation. I'd like to introduce Dr. Seung-taek Kim, the head of Zoonotic Virus Lab, Institute Pasteur, Korea. The title of his presentation is Identification of Antiviral Drug Candidates Against SARS-CoV-2 from FDA-Approved Drugs. Please join us to welcome Dr. Seung-taek Kim.
Well, thanks for inviting me in this interesting meeting. My name is Seung-taek Kim, and I'm working at the Institute Pasteur, Korea. Today, I am going to talk about the identification of antiviral drug candidates against the SARS-CoV-2 from FDA-approved drugs, as you can see from this slide. All right, this slide shows the current status of global COVID-19. We have more than 50 million confirmed cases and more than 1 million deaths worldwide. Well, the major risk factor for COVID-19 is age, and many COVID-19 patients, well, approximately 80% show mild diseases, but the others, about 20%, experience severe ones, even including deaths. The causative agent of COVID-19 is a novel coronavirus, which is named as SARS-CoV-2. And this coronavirus is an RNA virus, and it is very similar to the bat coronavirus. Okay, this slide shows the life cycle of coronavirus. Coronavirus is an enveloped spherical virus with the positive sense and the single-stranded RNA genome. Okay, let's look at the phylogenetic tree of coronaviruses. There are 4 coronavirus groups, alpha, beta, gamma, and delta. So far, a total of 7 the human coronaviruses have been reported. I marked these 7 the human coronavirus in the 2 different box colors. In general, the coronaviruses in blue boxes cause mild diseases such as common cold, but the coronavirus in the red boxes cause severe diseases such as COVID-19, SARS, and MERS. In terms of viral genome sequence identity, SARS-CoV-1 and the SARS-CoV-2 are very similar to each other by approximately 80%. Okay, before we talk about treatment options for COVID-19, we have to consider the clinical course of COVID-19. Okay, one thing that distinguishes the mild from severe cases is that how promptly innate immune response is initiated. Well, as you can see here, the innate immune response is delayed in the severe cases. For the patients with the mild COVID-19, no specific treatment is required. However, for the patients with the severe COVID-19, we have to choose treatment options very carefully, depending on which phase the patients are. The early phase is a viral phase in which the viral replication dominates. However, in the late phase, the excessive immune response dominates. Therefore, this phase is called inflammatory or a cytokine storm phase. In the viral phase, antiviral drugs should be effective. They play diverse roles in different steps of virus life cycle. And for example, convalescent plasma and the monoclonal antibodies would work by inhibiting interaction between viral spike protein and the host receptor protein H2. So these drugs act by inhibiting viral entry. For coronavirus entry, another protein, TMPRSS2, is required. This is the serine protease and facilitates virus entry by priming a spike protein for fusion event between the virus and host cells. This TMPRSS2 protein can be inhibited by camostat or nafamostat. Lastly, the remdesivir, which is the most famous drug in the current pandemic, is an inhibitor of RNA-dependent RNA polymerase. Therefore, this drug inhibits viral genome replication. However, in the cytokine storm phase, the drugs which can reduce excessive immune response should help. Steroid drugs are good drug candidates, and for example, dexamethasone has shown proven efficacy in the clinical trials. This slide shows clinical trials for COVID-19. More than 2,000 trials have been and are being conducted globally, and most of them are trials for drug repositioning. So in the pandemic situation, a new drug development is not really practical because generally it takes more than 10 years and needs a lot of investment. But even after these efforts, the success rate is still less than 10%. Therefore, something else has to be done in the current pandemic situation. So in order to respond quickly to the public demand of therapeutics for COVID-19, our lab started drug repositioning study early this year. Compared to the new drug development, drug repositioning has several advantages. Since the drugs are already proven in terms of drug safety and the pharmacokinetics, etc., once the antiviral efficacy is demonstrated, the drugs can skip the preclinical and the phase 1 clinical trials and then directly can be tested in phase 2 or phase 3 clinical trials. If those trials are successful, the drugs can be used for patients sometimes after emergency use authorization. All right. Even before obtaining the SARS-CoV-2, we started to work with the SARS-CoV-1 because we thought the drugs that show antiviral activity against the SARS-CoV-1 would have the similar antiviral activity against the SARS-CoV-2. Okay, this figure shows the procedure of our screening experiment with SARS-CoV-1. Briefly, the virus cells were seeded in the 384 well plate, and 24 hours later, the cells were treated with each drug and infected with the virus. Another 24 hours later, the infected cells were fixed and stained with immunofluorescence. These cells are now ready for scanning and image processing. Those processed cell images are converted to these dose response curves. The blue lines indicate inhibition of virus replication in the presence of increasing concentration of the drugs. The red lines indicate cell viability. A good drug candidate should have a low IC50 value with low or no toxicity. Okay, we started the screening experiment with 3,000 FDA and IND approved drugs. Out of these, we selected 35 potential antiviral drug candidates and later added 13 drugs which were suggested by infectious diseases specialists. So this slide shows our drug repositioning timeline. We received SARS-CoV-2 from Korean CDC on February 17. Before receiving the virus, we were screening the drug library with the SARS-CoV-1, as I told you in the previous slide. Well, as soon as we received the SARS-CoV-2, the virus was propagated and for experiments, and we developed antiviral screening assay against the SARS-CoV-2. Once we established the procedure, we immediately assessed the antiviral efficacy of the pre-selected 48 drugs and finally identified 24 drugs candidates against the SARS-CoV-2. Well, this whole process was completed within 4 weeks after receiving the virus. And a week later, our manuscript was posted in bioRxiv. Well, as you can see here, this manuscript is now published, and to the best of our knowledge, our paper was the world's first drug repositioning study with this scale of screening. Okay, from now on, I'm going to talk about the results. This slide shows the results of the reference drugs such as remdesivir, chloroquine, and lopinavir. Well, as you can see, our screening is an image-based antiviral assay. The red signals are coming from the nucleus of the host cells, and the green signals are from the viral protein. As the drug concentration increases, green signals disappear. These image data were converted to these dose response curves. For example, IC50 of remdesivir was 11.42 micromolar. And actually, we assessed the antiviral efficacy of the other 45 drugs against the SARS-CoV-2. And I'm going to talk about a few of them now. All right, the first drug that we were interested in was cyclesonide. You can see the beautiful image strip as well as the dose response curve. The IC50 was 4.33 micromolar, so approximately a half of IC50 for remdesivir. This drug is an inhaled steroid drug and originally developed for asthma treatment. Well, interestingly, when we were preparing our manuscript, another similar result was submitted by a Japanese group, and they reported that NSP15, which is a viral end-ribonuclease, might be the target of cyclesonide. So based on these two facts, we expect that cyclesonide may play dual roles in the treatment of COVID-19. First, as a direct-acting antiviral targeting NSP15, and the second, as antiviral anti-inflammatory steroid drug. And based on our results, a phase 2 clinical trial is being conducted in Korea. The next drug that we were interested in was niclosamide. The IC50 was very low, about 0.28 micromolar, which is much lower than the IC50 of remdesivir. This drug is actually used as an antihelmintic drug. But interestingly, over several years in the past, numerous laboratories in the world have reported its broad-spectrum antiviral activity, even against SARS and MERS coronavirus. And coincidentally, its mechanism of action was revealed last year. Using the MERS coronavirus, a German group found that niclosamide enhances autophagy, which reduces replication of MERS coronavirus. So far, well, so good, but this drug is not absorbed very well. So if this drug is developed as an antiviral drug against SARS-CoV-2, this problem has to be solved. Well, anyway, after releasing our results, we received numerous responses from all over the world. Well, as far as we know, now several companies are trying to develop methods for effective delivery of niclosamide, and a few phase 1 clinical trials are being conducted in Korea and in foreign countries. Well, so far, I have talked about the results obtained from viral cells. These cells were originally isolated from African green monkey kidney, and one of the very frequently used cell line in virus research. However, drug susceptibility can be altered in different cell lines, so we try to develop an image-based antiviral screening method with another cell line, and we use the human lung cell CALO3. We think this cell line would be physiologically more relevant for drug testing. And though these cells were not easy to work with, we successfully developed the screening protocol. You can see the nice image strips and dose response curves of the reference drugs in this slide. Well, since the CALO3 cells do not grow very well, we focused our antiviral screening on the 24 drug candidates that we identified in the viral cell screening. Obviously, IC50 values in CALO3 were different from those in viral cells. And for many drugs, IC50 value increased in CALO3. In other words, the drugs became less potent in CALO3 cells. Well, specifically, the IC50 of chloroquine is increased by 10-folds. And this was also demonstrated by another study conducted by a German group. And now we know chloroquine and hydroxychloroquine do not help COVID-19 patients based on the results from several clinical trials. Well, however, for some drugs, IC50 rather decreased in CALO3 cells. Well, remdesivir was one of them, and actually IC50 of remdesivir decreased by 10-folds in CALO3 cells. Here, we need to pay attention to two other drugs. These are nafamostat and camostat. Look at these numbers, 0.0022 micromolar and 0.187 micromolar. In CALO3 cells, their IC50 values decreased tremendously, which means these drugs can be very potent antiviral drugs in SARS coronavirus 2 infected the human lungs. Although nafamostat and camostat were not selected as the 24 potential drug candidates in our viral cell screening, we included these drugs in calorie cell testing because of the earlier report from Germany. This paper shows that the TMPRSS2 can be a target of antiviral therapy and the demonstrated antiviral efficacy of camostat in vitro. In addition, very interesting case reports were published by Korean and Japan the clinicians regarding nafamostat as antiviral therapy for COVID-19 patients. So then the question is, why is there a substantial difference in between these two cell lines? Well, fortunately, we have some clues where SARS-CoV-2 seems to enter the cells in two different ways. And these pathways are likely determined by the expression of TMPRSS2. In calorie cells, TMPRSS2 is expressed, and this protein primes viral spike protein before fusion occurs between viral membrane and then plasma membrane. However, in the viral cells, the TMPRSS2 is not expressed very well, and the virus enters the cell via endocytosis. In this case, the fusion occurs between viral membrane and the endosomal membrane. Therefore, in calorie, the TMPRSS2 inhibitors, such as nafamostat and camostat, are effective. But in vivo, chloroquine and the hydroxychloroquine are effective because these drugs inhibit endosomal acidification, which is necessary for fusion to occur. So in this slide, I compare the nafamostat dose response curves between the viral and the calorie cells. You can see the very low IC50 of nafamostat in calorie cells. Nafamostat was originally developed as a drug for pancreatitis, and it is also used as an anticoagulant. For numerous COVID-19 patients, blood clotting is a serious concern. So we expect nafamostat would play dual roles in the COVID-19 patients. First, as a viral entry inhibitor, and the second as an anticoagulant. So based on our results and others, several phase 2 clinical trials are being conducted in Korea and foreign countries. Okay, this slide is for camostat. Again, I compared the camostat dose response curves between viral and the calorie cells. IC50 in calorie was very low, but less potent than nafamostat. Camostat is also a drug for pancreatitis, and it should work as a virus entry inhibitor. One major advantage of camostat is that this drug is orally available. However, nafamostat should be administered by IV injection. So we expect that camostat would be good for patients with mild cases, and nafamostat would be good for patients with severe COVID-19. So based on our results and others, several phase 2 clinical trials are being conducted in Korea and then foreign countries. So in summary, we developed image-based screening for both viral and the calorie cells. In the beginning, approximately 30,000 FDA and IND approved drugs were screened to identify antiviral drug candidates against SARS-CoV-1. Out of 48 drugs that we selected from SARS-CoV-1 screening, we identified 24 drug candidates against SARS-CoV-2. And among these, cyclosonide, nafamostat, and the camostat are being tested in phase 2 clinical trials, and the niclosamide is being further developed for better absorption in the body. All right, many people helped us in this investigation, our team members and other people in our institute. SARS-CoV-1 was kindly provided by Malik Peiris at the Hong Kong Pasteur, and I also would like to thank Korean government for supporting our research and the Korean Society of Infectious Diseases for their kind advice. Well, thank you for your attention.
Thank you for a nice presentation, Dr. Kim. We will move on to our second presentation. The presentation will be given by Karin Lore, the professor of Karolinska Institute. The title is Preclinical Testing of COVID-19 Vaccine Candidates. Please join us to welcome Professor Laura.
Thank you. I assume that you see my slides now. So thank you for this invitation. I will talk about testing of COVID-19 vaccine candidates and some work that we do. Under normal circumstances, we test a number of different vaccines to several different pathogens, but during this pandemic, our priorities have at least temporarily shifted some. So this is to put things into perspective of the procedure of vaccine development that starts with identification and screening of relevant vaccine compounds that could go into a vaccine, and then preclinical testing in smaller animal models to larger animal models, followed by phase 1, 2, 3 trials, and then submission for approval. And I wanted you to take a look at this timeline. 10 to 15 years is normally what it takes to develop a vaccine, and that is when it's a successful process. There are several vaccines that we have worked on for many more years, for HIV more than 30 years, for example, and we still do not have an effective vaccine. So the speed that is going on right now with developing a COVID-19 vaccine is really special. And this is an exciting time, both that vaccinologists in the academia can work together with the vaccine industry, and expertise and experience that have been built up for years can now be put into play and be utilized. But it's also a bit scary that with this speed, there may be potential risk that we will have partly ineffective vaccines or even worse, vaccines with rare side effects that we will not see until we start to mass vaccinate. So my group consists of immunologists. So that means that we have a number of laboratory assays to measure the magnitude and quality of vaccine responses and different aspects of vaccine responses. We are not making vaccines. That is an expertise in itself. The vaccines that we're testing comes from the vaccine industry in most cases, and sometimes also academic partners, often protein designers. All the vaccines that we work with are in late stage development. So that means that they have already been filtered and selected because they show very promising results. They have already gone through rodent testing, for example. So that's when we come in. So we test the vaccines right before they go into clinical trials. We test them in non-human primate rhesus macaques at the Karolinska, or in phase 1 clinical trials. So live vaccines are, in general, the best vaccines. They most closely resembles the real pathogen. And the yellow fever vaccine is the world's best vaccine, one shot and lifelong immunity. And one live vaccine that we're currently testing is the Bordetella pertussis vaccine that's given directly nasally into the nose. And we're doing this test together with the Public Health Agency of Sweden, since the current pertussis vaccine is not completely effective. But for safety reasons, live vaccines cannot be developed to a number of pathogens that we need vaccines to. For example, HIV can never be built on a live attenuated vaccines. So many of the vaccines that are being developed today are based on protein subunit vaccines, meaning that you take one or selected proteins of the pathogen and make that into a vaccine. And for this, the nanoparticle technology has really become popular, and I would say one of the trendiest technologies today that's being tested in vaccine development. And two vaccines that are licensed with this technology, the virus-like particle technology, is the hepatitis B and HPV vaccines. And they are really strong vaccines and have really turned into blockbuster vaccines. So of course that has spurred the development of vaccines based on this nanoparticle technology. And some vaccines that we are working with, there are nanoparticle vaccines. We are malaria vaccine that we just completed testing. We're working on an RSV vaccine intended for pregnant women and elderly, and an HIV vaccine. And all these vaccine candidates are being tested in non-human primates at the Karolinska, so they are not yet in clinical testing. And these studies are supported by the Gates Foundation that has as one of their missions to eradicate some of the global infections. Then we have the mRNA or nucleic acid vaccine platforms based on mRNA and DNA that I've heard a little bit about this morning. There are no licensed vaccines yet based on this technology in humans, so that's important to remember. But this is a quite exciting technology that could have the competitive advantage of that can be produced very rapidly. As soon as the sequence is identified for a pathogen, we can theoretically produce an mRNA vaccine, for example. And it's believed that this is highly scalable and it may also be quite affordable. We'll see. So for a number of years, we have worked with Mrna to test an pandemic influenza vaccine, and we're also working with CureVac in Germany. Mrna is in the US, as you may know. So of course, this was a very relevant vaccine platform when the pandemic started, because as soon as the sequence was identified for SARS-CoV-2, a vaccine started to be produced. So this is how the landscape looked before the pandemic, and after the pandemic, we had to shift gears a little bit, at least temporarily. So these are the vaccines that we're testing today. We're continuing to work with CureVac, and we test their mRNA vaccine to SARS-CoV-2. We're working with Novavax that has a nanoparticle vaccine with a matrix M adjuvant, which is an adjuvant that's actually developed in Uppsala in Sweden. And we're also testing a whole virus inactivated virus vaccine developed by the US Navy. So these are studies in non-human primates again, while, at least for Novavax and CureVac, the clinical trials are already going on. So again, this is a difference compared to how it normally works, that all the animal testing is done before the clinical trials are starting, but as it is now, everything is going on at the same time. So most COVID-19 vaccines are based on the spike protein. So that is the protein that sits on the surface of the virus and is used by the virus to infect cells. And the spike protein is relatively large, and it has one receptor binding domain, which is likely very important because that is what this virus is using for infection, and it's likely important to block that particular part of the spike protein. So the spike protein as a vaccine can come in different forms. It can come as an mRNA sequence, as I discussed, and these are the main players in frontline clinical development, but that's not to say that there are many smaller companies also that are pursuing different versions. We've heard probably everyone in the news about Moderna's and Pfizer-BioNTech's mRNA vaccines that look very promising. And we also have adenovirus vectors. They're also based on a sequence for the spike protein. And just around the corner after these front runners, because these are a little bit ahead, then we have the protein subunit vaccines that is in many cases are nanoparticle based, like the Novavax, for example, and they are based on a recombinant spike protein. And I'm not an advocate for any of these platforms. I think they have their advantages and disadvantages, all of them. But I think one thing that is important to remember is that protein subunit vaccines we have used, and we have licensed for other pathogens, and they are generally very good vaccines. They use high levels of neutralizing antibodies, and they're in most cases safe. So that is something to remember here, that when we go into a potential mass vaccination, that these platforms are not yet approved for other pathogens, so we have less experience what's going to happen here. And as I said, we work with CureVac and Novavax. So this is an example of a vaccine study outline. As I said, the clinical studies are already going on. So what is the purpose then of doing non-human primate testing at the same time? Well, it's actually critical, because we have to now turn every rock, learn as much as we can of how these vaccines are working and how they interact with the immune system and any potential side effects and all the information we can get. So this is of utmost importance. So a vaccine study is often performed in the way that we immunize, exactly like you would do in a clinical study, and then you follow. And we have the advantage of working for a long time, so we can look at durability of the responses for a long time with regular blood draws. We can draw bone marrow to look at resident bone marrow plasma cells. They're long-lived antibody-producing cells. And in terms of COVID-19, we can look at mucosal tissues that are relevant for this infection. So nasopharyngeal aspirates or bronchial lavage, meaning in the nose or nasal cavity, and also deeper down in the lungs, to see if we have antibodies there or T-cell responses with this vaccine. We can also perform a virus challenge, which is the ultimate proof of that a vaccine will work. Rhesus macaques are susceptible for SARS-CoV-2, but it's not a perfect challenge model because they don't get very sick, but they are susceptible. So one can measure prevention and protection from developing pathology. And as I said, we have a number of methods that we are using. Safety is always important. We look at innate immune activation, the responses you get immediately after, so a day after vaccination, because that may be absolutely critical how you imprint the vaccine response. Most vaccines, if not all, work via induction of antibodies, so we can really penetrate the B cell populations and look at the different kinds, germinal center B cells, different populations, and also the type of antibody response you get. We can look at different tissues, for example, look at what happens in the lymph nodes that are draining the vaccination site. This is before vaccination, and this is 2 weeks after vaccination. So without knowing much about germinal centers, you can see that there is a drastic difference 2 weeks after vaccination, that you're developing a B-cell response to this vaccine. We can also sort antigen-specific B cells, and we can sequence them to look at maturation of the response. We can look at how many different clones there are, and we can identify and clone antibodies. So we can produce antibodies and analyze them. And theoretically, if a really good antibody is discovered, this antibody could be tested as a therapeutic antibody in the future. We can also look at T-cell responses. CD8 T-cells may not be the correlate of protection, but in any event, you will need CD4 T-cells to build up a good B-cell response. And we can also do biodistribution studies. So that means where does the vaccine go and how long does it retain in the body after you vaccinate. So that is an important safety measure. This is just an example of how it can look. Spike-specific IgG levels. You vaccinate and you see an increase of antibodies being developed. Boost and you see a further development, and then you will measure the waning, because you will always see a waning, and how long can we actually have well-detectable antibodies. If we compare those antibody levels to COVID-19 patients, for example, this is the levels we have prime and boost in these monkeys, you can see that after boost, they're lining up in the higher range of COVID-19 patients. And these patients represent a range of disease severity. So many of the severe disease patients are up here and the milder are here. So with this vaccine, we seem to induce a good antibody response. As I said, we also look at B-cell and memory B-cell responses, and this is important to understand how long we have memory B-cells around, and because that could be a measure of our chances of responding upon re-exposure to build up a memory response and thereby be protected. So we can label this memory spike-specific memory B-cells, and we can enumerate them over time, and again compare them to what we would see in COVID-19 patients. So this was just a short summary of the field and what we do. I think that there are several promising vaccine candidates underway, and I'm very optimistic that we will have multiple vaccine candidates that are good. And our studies have shown that we induce high antibody titers, and that you see with several other vaccine candidates as well. There is also a memory B-cell pool being developed, so that's good for potential re-exposure. As I mentioned, to know more about these vaccine candidates that are already in clinical trials and maybe filed for approval, I think we still need to understand these vaccines so we don't get ourselves into a position where we start to see side effects. We had a very unfortunate event here in Scandinavia, 2009, where we had mass vaccination for the swine flu, and we had increased cases of narcolepsy. So we should do everything we have in our power to make sure that we don't run into similar problems. With all the vaccine candidates that will be developed, I think that it would be important to consider which type of vaccine will go to which group in the human population, because that is really something I think one should consider. And with that, I think I would close. I think my time is up, but thank you very much for the invitation to speak in this workshop.
Thank you, Dr. Laura, for sharing very important piece of your work. Let me go on to the third presentation by Bang-Lin Song, moderator myself. And the title of the presentation is Vaccine Platform in the Post-COVID-19 Era. Hello, everyone. I'm Bang-Lin Song, a professor at Yonsei University and the Director General of Vaccine of the Technology Alliance, Korea. I would like to use this privileged opportunity to address to a vaccine platform in the post-COVID-19 era, with the hope to establish the collaboration between Korea and Sweden. Let me start with a brief introduction of vaccine and special consideration akin to the Corona-19 vaccines and vaccine strategy in the post-Corona era. And I would like to end my talk with my personal proposal for the mutual collaboration. The benefit of vaccination cannot be understated. Prime example is smallpox. One of the successful examples of the fighting against infectious disease is the smallpox vaccine. Due to this accomplishment, the virus is eradicated in 1977 as announced by WHO. Another successful example is polio, which is near elimination except for the sporadic outbreak in the tropical area. One of the newer successful examples of the vaccination is meningococcal disease. There are at least two different types of serogroups. Now, after the vaccine against serogroup C is developed, the instance is greatly dwindled, whereas serogroup B still persists, requiring another type of vaccine and serogroup B. Despite this successful example of vaccine development, the size of vaccine trial is is ever increasing, creating high barrier for small and mid-sized companies. One example is Rotatech that requires about more than 70,000 people recruited in phase 3 clinical trial. However, the emerging virus infection is more than 10 different viruses. COVID-19 is just the tip of an iceberg, and WHO blueprint list of priority disease stipulates vaccine development of at least 10 different emerging viral infections: MERS corona, SARS corona, Zika, chikungunya, dengue, Crimean Congo hemorrhagic fever, Lassa fever, and Ebola virus. However, the conventional vaccine paradigm, based upon 3I principles, isolate the identifying the infectious disease and inactivation followed by injection. This conventional vaccine paradigm requires at least 10 or 15 years of development. To switch from this conventional paradigm, new approach is beginning to emerge: conjugate vaccine, reverse vaccinology, structural vaccinology into virus-like particles. However, this new development of vaccine strategy is just short of the global spread, the catching the global spread of the COVID-19. As of last month of this year, COVID-19 caused infection over 30 million people, causing 1 million deaths, with the death rate of 3 to 4 points of the death rate. We should remember the COVID-19 is a genetic variant of the SARS-CoV-1 that appeared in 2002. So it's about 17 to 15 years before the global spread of SARS-CoV-2. So if you use this lesson, then we should be aware that the MERS coronavirus that appeared about 5-6 years ago may develop into a MERS-CoV-2 within 5 or 10 years. We should be well prepared for this type of scenario. There are 2 different pillars for vaccine development. It should be safe and efficacious. However, the COVID-19 outbreak requires another pillar of development, that is speed. So based upon conventional 3I principle, the vaccine development is slow, the cost is very high. To adapt to new normal situation, there are various types of new vaccine platforms: DNA vaccine, RNA vaccine, vector vaccine. These platforms are being adopted by the major companies like AstraZeneca, Pfizer, GSK. And I would like to stress that one additional new normal type of vaccine development is a virus-like particle, VLP vaccine, which can be produced in a very short period of time in a low-cost manner. Safety issue is the most important for the coronavirus vaccines. Therefore, the safe vaccine platform, DNA vaccine, mRNA vaccine, has been adopted as a safe vaccine platform. However, we should not forget the safety issue still lingers on the virus itself. For instance, antibody-dependent enhancement disease can cause a problem. For instance, after vaccination, rather than providing protection, the antibody may increase the disease phenomena. The safety issue lingers around COVID-19 vaccine, although so far the phase 3 trial, until the phase 3 trial, there is no manifestation of the safety issue. That's good news. Let me address to the VLP vaccine. Virus-like particle is morphology-wise is like the virus. However, it does not have a genome. Therefore, it is not infectious. After vaccination, since it resembles the infectious virus, after vaccination, it elicits robust immune response, provide a good protection. This virus-like particle has been produced through the eukaryotic system, baculovirus, yeast. One of the successful examples is the Cervarix or Gardasil for the prevention of the human papillomavirus infection. However, E. coli system provides much faster means of the vaccine development. The speed, scalability, yield is very, very high as compared to the mammalian and bacular system. However, the E. coli system is so simple and does not allow the assembly of virus-like particle. That's the main issue of the technical hurdle. If you achieve a technical breakthrough, you can use the bacterial system for the delivery of the low-cost vaccines in a speedy way. This technical breakthrough has been achieved through the past collaboration between Korea and Sweden, and the Yonsei University, where I'm affiliated with, and the Stockholm University. And there are various papers that co-authored between these two institutions. The virus-like particle, for instance, norovirus, has 180 monomers. Regular assembly is required. That is very, very difficult task. And the regular assembly of the more than 180 blocks required piling up of each antigens in a very kinetically controlled way. If you are so haphazard, it will crumble down. Therefore, this monomer antigen must keep the distance between them. So-called social distancing principle applies to vaccine design, especially with the assembly of virus antigen. I would like to mention a co-authored paper between Sweden and the Yonsei University, a social distancing measure governing the whole proteome that exactly applies to vaccine design. One of the successful examples is shown in norovirus. Norovirus vaccine, VLP vaccine, requires assembly of the 180 different monomers. This is one of the successful examples, and this technology is transferred to a major company in Korea as under commercial development. One example of the importance of production speed is shown in influenza vaccine. The conventional egg-based vaccine requires 6 months. Newer cell culture method requires 4 months. However, bacterial system, if successful, can be developed within a month with Operation Warp Speed dedicated from the US government. I'd like to mention a subgenomic assembly, a so-called pentameric-based assembly that could be applied for the various type of vaccines applied to the flaviviruses, including dengue virus, West Nile, Zika, and Japanese encephalitis virus. That uses a cholera toxin B as a scaffold for pentameric assembly. I should not forget to mention an eminent scientist in Sweden, Ian Holmgren, who is affiliated with the University of Gothenburg. He used to be the scientific advisory board member in International Vaccine Institute in Korea. And I have established long friendship with Dr. Holmgren. And through this collaboration, his inspiring method for cholera toxin B at the scaffold pentameric assembly was instrumental for the vaccine prototype, the dengue, West Nile, and Zika viruses. So, the same vaccine assembly platform, virus-like particle, nanoparticle vaccine, are applied to norovirus, FMDV virus, poliovirus, and flavivirus, including dengue, Zika, Japanese encephalitis, and as a fast delivery platform of influenza vaccines. And the emerging virus, as a prime example of emerging virus, Nipah virus. All this type of the same principle of the virus-like particle and vaccine provide a platform to approach to various type of viruses. Now, the scientists is beginning to worry about the twin endemic situation, corona and the influenza. The conventional influenza virus should be developed every year and vaccinated annually. It's a kind of moving target because at the end of the year, there are genetically variant type of virus appears. It's a kind of moving target. The vaccine approach for the seasonal influenza vaccine is very ineffective and costly and requires time. Nowadays, scientists are working on the development of universal vaccine. It by one shot protects the current virus and also other genetic variants that is to come. So the shift in vaccine paradigm is developing universal vaccine, and that applies to coronaviruses. Now, the influenza and corona may circulate closely, and there is a possibility the co-infection of these two viruses may aggravate the situation, calling for the universal vaccine, the twin endemic vaccines. And I have developed a universal influenza prototype as exemplified by the recent article, The Quest for a Truly Universal Influenza Vaccine, in my laboratory. Now, our task is to develop a common vaccine platform assuring the fast delivery of the low-cost manner, which could be applied to the various vaccines of various viruses as stipulated in the WHO blueprint list of priority disease. I would like to close my talk with my personal proposal on the collaboration on the delivery of the low cost of the pandemic vaccines. For instance, as an alternative to cell culture, FMDV noroviruses, and as an affordable premium vaccine, HPV, as a fast delivery of the pandemic influenza vaccines, and the vaccines against emerging and neglected disease, and the improvement of the pre-existing vaccines, and the post-eradication management of the polio. Based upon a long history of collaboration between Korea and Sweden, I would like to propose the funding body, the Bill and Melinda Gates and CEPI, and the newly formed Korea initiative, the Global Fund, Right Fund. They actually invite proposal for international collaboration for the infectious disease. I'd like to close my talk with brief mentioning on the vaccine innovative technology, Vital Korea, is a kind of a Korean government research initiative for the next 10 years with the amount of $200 million worth of investment from the Ministry of Health and Welfare. There are two mandates to achieve: to increase the vaccine self-sufficiency in Korea, allowing the vaccine domestic production, and also developing vaccines on global unmet needs. This is conventional two different mandates. Now I am negotiating with the government to add one more mandate to establish a common vaccine platform against the post-corona era, to be delivered to the low middle income countries of the low-cost vaccine in a speedy delivery. And Korea has an advantage of establishing International Vaccine Institute, which has been in Korea for now 23 years. And with the IVI as a collaborating partner, we can establish a international collaboration through the aid of the international funding bodies, CEPI and BMGF and Right Fund. Thank you for your attention. I think this pretty much wraps up the first session, and we can have a short time to discuss for the presentations given. And if you have any questions from Sweden and Korea, please feel free to participate. So 3 presentations given in this session, as well as the plenary speech by Nary Kim. All of these talks are within our discussion. If not, let me start with my question to Dr. Lore. Do you hear me?
Yes.
Okay, all right. Thank you, very nice presentation. Could you briefly mention on your VLP platform what type of system you are using and how many different vaccine targets you are addressing with your platform?
Sorry, so you mean how many spikes there are sitting on each nanoparticle?
You mentioned several different platforms, live attenuated, VLP, mRNA, and adeno. And since I speak about the VLP, I'm interested in your VLP vaccine platform in Karolinska Institute.
Yeah, so it depends on, I mean, again, we are not making the VLPs. That's the work of our collaborators, which are usually vaccine companies. But it depends on which VLP or nanoparticle platform. So that can be a range of 20 to 300. So it's quite a range of vaccine antigens that can sit on one virus particle or nanoparticle. And this can be in a very organized way. So in a way you really have like a self-assembling, directly designed way, so you know exactly the stoichiometry of these virus particles, or it can be that they're packaged into a polymer, which is then a more random distribution. And I think that there are benefits to both of them, because it's not necessarily so that you wish to have as many proteins as possible on one virus particle, because what can happen then is that you actually mask important epitopes. That's something we have seen. That's, you want to have a good balance of that the B cells with the B cell receptors can get into, for example, an epitope closer to the base on a virus-like particle. So, yeah, so...
Okay, thank you. Yeah, Dr. Larry Kim.
May I ask a question too? I'm an RNA biologist, so I'm really interested about the mRNA vaccines that you're working on. You've been working with Mrna and CureVac, so I was curious what's your insight as to the this early stage of mRNA vaccines. I'm sure there are a lot of rooms for further improvement, and I was wondering what you think is the current limitation and what are the challenges that need to be made in the future if you want to develop mRNA
yeah, I mean,
platform.
this is an excellent question. that I think a lot of people think about. I think one very urgent issue to resolve is storage of the vaccine. And now it looks like the storage is actually not necessarily, even though the Mrna's vaccine is not necessarily needing -80 cold chain, which was first what was told, that it can actually be stable for several days in a fridge. Because distribution of such vaccines, our primary care units do not have -80 freezers.
Sure.
So it wouldn't work to have vaccines with those requirements. And I know the mRNA vaccines in general have issues related to this that they have to deal with. So that just from a practical point of view, that becomes a large issue. When it comes to a medical issue, I think that, there are some side effects that have been reported. These mRNA vaccines induce a very high type 1 interferon response, which is likely a good thing that they induce a vaccine response in an efficient way, and maybe in particular to viruses, since type 1 interferons are a part of the natural response to viruses. But one has to be careful that this is not too much type 1 interferon, because that's going to be detrimental and gives side effects. So I think that we will see when more and more people are getting the vaccine that this is actually not too much of innate immune activation. Because if you go through the literature of the clinical trials, you'll see that there is a quite high number of side effects reported.
If the I saw that the doses of these mRNA vaccines are over 25 microgram in most cases. Do you think if you can reduce the amount of mRNA for the vaccine, you can reduce the side effects? I mean, if you can increase the duration and gene expression levels of mRNA, then I guess you can easily reduce the amount to a substantial level.
No, again, this is an excellent question and point that you bring up. One issue is that the spike protein is very large. So if we go too low down in dose, we get very low translation efficiency. And this is also what, for example, protein producers see when they just produce the spike protein as a recombinant protein in the laboratory, that the yield is very low. So that is a challenge, that the spike protein is very large, and therefore it's a little harder for the targeted cells to produce or have a massive production of this protein. So therefore, I think it's a limit of how low you can go in terms of dose. But I definitely agree with you that that's where you would start to look, if you could go down to dose and get rid of the side effects. And also with lower doses, we can produce more vaccines and also distribute to countries that may not afford to pay high costs for vaccination. So that is definitely a large incentive to try to investigate this further. And having mRNA vaccines scalable and produced at a low cost, that was sort of one of the selling arguments in the beginning for mRNA vaccines. And it will be interesting to follow if this actually will be the case.
Thank you.
For the interest of time, I think we had better close the first session at this moment and go on to the next session. And I would like to thank for all the participants from Korea and Sweden, and I hope this provides the first step for the collaboration for the mutual interest in the long run. So let me move on to the second session. We have three more speakers from Korea and Sweden. The first speaker in this session is Stefan Lundström, Chair, Chaired Professor in Fluid Mechanics of Mrna University of Technology. He presents effect of face masks during airborne pandemics from fluid mechanical aspects, a collaboration between 4 Swedish universities. Please welcome Professor Lundström. Please.
What I present today is a project that we have together 4 Swedish universities, and I will also just present what we will do and the tools that we will be using, because the project has just started and it's sponsored by the Swedish Research Council. Just before presenting the project, I will just notice that my main colleague in Korea is Wook-Rol Wang. He's Professor Chair of School of Mechanics and Aerospace Engineering in Yeosan National University in Jeonju. And we work in different areas within fluid mechanics, well, not directly within this project that I will present. But I really enjoyed my visits to Jeonju and Korea. The theme for the project we have on these fluid mechanics aspects of face masks are from 4 Swedish universities and includes these persons. And especially mention Johan Restedt in Lund, who put together or initiated the collaboration that was actually this project that we started. And it's actually based on work that we have done together with the Swedish industry in multi-phase flow, including industries such as Tetra Pak and AstraZeneca, for instance. The purpose then with our work is to reveal how liquid particles behave close to people. So we want to look quite close near the mouth and nose, for instance, wearing face masks or not when inhale, exhale, and include cough and sneeze. And this is also done by other researchers all over the world in parallel, we can say. And then within a year, then we should, from a fluid mechanical point of view, be able to give guidelines of the use of face masks to prevent spreading of virus such as COVID-19. We have 4 work packages. This animation that is led by myself, simulation of droplets flow dynamics, and here we said that we will work close to the mouse, but actually in this project we will also look at particles moving further away from humans than breathing or coughing or sneezing. We're also, this is simulations, and also we will do simulations of droplet and aerosol transport from mask material, and then there is experimental validation, and this is done at the different universities then as shown here. This is also the steering group that we have in this collaboration. So regarding this simulation, this is a rather interesting area for society in general. So myself has been on Swedish television showing a little bit the complex of the flow when you inhale and exhale. For instance, it is very difficult to put out the candle by inhaling. Like this is nearly impossible, but of course exhaling is quite easy. So there's a big difference already there. And then if you have a face mask, it will of course be an even more complex flow situation. Then going more into the real work, so to speak, we have the simulation of droplets flow dynamics, and here is done to look in larger environments like a room where I'm sitting, or it could be a bus or office, or it could be a shop or drugstore. And here it is the effect of face mask on safe distance, droplet distribution, and droplet and vapor dynamics. And connecting to the talks here at the workshop so far, one of course interesting subject is this how long viruses can live in different environments, because we can maybe predict where droplets may be having viruses can be at a certain time, but then of course do they still have virus in them? Yes, there will be validation here, because that's you always have to do when you do numerical simulations, because they're always approximations. And we will develop a baseline case together between the different universities. And also the model that developed then at Chalmers with the flow through the mask will also include non-Newtonian effects of the liquid, and that is something that people then cough out or sneeze out, so to speak. Here are some preliminary results of this work package that we have. This is then a study of just a space that is mainly 3 by 3 by 3 meters. And this is this kind of numerical mesh that is used in computational fluid dynamics. It's often important that this mesh is done in the right way. And here's then just one example of particles moving, particles of different size are moving. And of course, the air of the coming up from human is hotter than the environment around. The air will eventually move up a little bit like this. But it's still a quite simple geometry. But what this result shown still is that there has been this kind of validation, that the experiment has been, that the simulations has been compared to experiments with a very good agreement as seen here. But this can then also be another kind of environment. But the 4% in this kind of simulations is of course very important that you know that they are relatively right. Work package 3 is about, as I said before, droplet and aerosol transport through mask materials. So here we'll formulate that must be a rheological description of complex fluids. This, as I said before, is spelled from nose and mouth. In the case of these various expiratory events, we will also study numerical spreading and absorption of complex fluid and aerosol in filter layers or protected masks. So we really will go into the mask material here. and the third step also to propose in innovative designs of these microstructures. And this will be done by a certain numerical method called Lattice-Boltzmann framework. We will also do it with conventional computational fluid dynamics, because one is for droplets and one is for aerosols. That is, a different size then of these droplets, so to speak. And we will do fibrous microstructures of real mask materials, and we'll show an example of that. I think I will take next slide. So here is then how the simulations could look like, where we use this Lattice-Boltzmann method, where we can really follow droplets moving through fiber networks, which in this case is a protected mask. And we can do this to a certain size of all these droplets. So what can the results be? Yeah, that could be how the permeability, the resistance or the ease of the flow, going how easy the air go through the mask, the probability of droplets as within the masks, as shown here is in this diagram, and also then how droplets move together or intercept, so to speak. The fourth work package is about experimental validation, and here we will do two use at LTU at my university. Then we will use two measurement techniques, and we will do real measurements, so to speak, and that is called interferometric particle imaging, where we can measure the speed of particles. We can also measure the size of them by this defocusing image of particles. And just the number of fringes then that we see are proportional to the size. And the size you can measure is, as it is now, 5 to 100 micrometers. And the other, the movie is showing then shadowgraph imaging, where we also can follow particles. And in this case, the person is saying P, the letter P, And this is well known that saying this kind of letters like P and B releases a lot of particles. This has been known for a long time, but we can also see here on these images a rather interesting mechanical phenomena of this fluid saliva that is released from the lips move away from this person. I can also say that the IPI is of course a really good method, but still we can just measure in a quite small volume. As maybe you can imagine that one of these, you can see the size of the particles here, and then you can see how big the size of window that you're measuring is. There will also be the complementary or similar measurements done at KTH, another university, physics lab, and here we can see some preliminary flow visualizations with smoke. There is a person that is breathing without mask, or is not a real person, but an imaginary person, we can say, and then inhaling and exhaling with mask. And for the exhaling case, we can see here that, of course, it's very important for how the mask fits to the face, that there can be leakages, and we not have this kind of filtering that we want to have in the mask. And inhaling is like coming from everywhere, so it's not so easy to do actual measurements of this. Anyway, for the future, both universities will focus on what happens very close to the mouth, because this can then be a good input to the simulations that we will do. So this is both these measurements that I showed at LPU, but then also KTH will do similar and complementary measurements. So this actually completes my presentation. I will just, as a final thing, show the X-ray tomography that we have done, or initial X-ray tomography that we have done for face mask, and this kind of our data we'll do for additional face mask, and will be used in this project. Right. Then thank you for your attention.
Thank you, Professor Lundström, for a very nice and intuitive presentation. Next will be the Korean speaker, Professor Reung Park. He is a professor at Aja University. The title is Global Clinical Big Data Research Network to Overcome COVID-19, Korea's Support. Please.
I am Reung Park from Aja University in South Korea. It's my honor to present today. I would like to present about the Korea's support on Global Clinical Big Data Research Network to overcome COVID-19. Some of you may know well about the CDM, but for someone who is not familiar with it, I will briefly introduce the basic principle of a common data model. Research using multiple healthcare data has many barriers, such as as a technical issues, including heterogeneity in data type and structure, data quality and volume, technical limitations, and legal issues, including privacy protection law, permission from data owner, and the human nature who do not want to share the data to others. To overcome these issues, okay, we convert all the structured EHR data into a common structure and common meaning. We call this common data model. And then the researchers outside of the hospital make analytic codes such as R or SAS or Python. Then the researchers send the research code into the hospital. Then the administrator in the hospitals just run the analytic code, and the resulting summary or the statistical result can get out from the hospital to the researchers. So the researchers only gathers the statistical analytic result, not the raw data sources. Then he can aggregate the result and get the he can know what he wants from the hospitals. So we call this distributed research network. By doing them, no personal protected health information can go out from the hospital, and the analytic codes can be used over and over. And the analytic code analysis over multiple, the researchers can do analysis over the multiple databases, and the analytic code can be reused multiple times. This is the national CDM project in Korea, started from 2018 to the 2022. Here, more than I'm in charge of converting more than 60 hospitals EMR data in terms of CDM. And on the top of the data network, we are also developing a platform named Findability that utilize all of this data. And also there are independent projects for companies who want to develop their own application service that works on the platform. The services include clinical trial support, CDSS, AI service for diagnosis and prescription, and biomarker discovery, like that. The Ministry of Health supports these 20 projects. 10 projects are for clinical researchers aiming to study public purpose clinical research using all of these data echo. Another 10 projects are for development of policy, guidelines, and technology enhancing the security and privacy protection of all these echoes. Moreover, there are more projects not listed in this illustration. Now, 61 hospitals, including 70% of tertiary teaching hospitals from all provinces of Korea, joined to the network. This is a diagram of the coordination platform named Findability. The researchers or company join to the Findability, and they can make their own analytic code, or they can request to the researchers who can able to make the analytic code. Then the analytic codes go into the hospital, and then it is run against their common data model, and the results are exported to the researchers. This is the CDM conversion process. I'm sorry for some Koreans. From the preparation and installation of hardware, software infra, ETL plan, ETL develop, and CDM conversion data quality. The whole process usually takes less than 3 months. Now we have the Findability, so I would like to show you how the Findability works. I would like to show you some aspect of the Findability in Korea. Findability.com here. Login. As you see, now there are 41 million patient data are now in CDM format, and now 28 hospitals CDM servers are integrated with this network, and now about 9 hospitals are in their conversion process, and now more than 4,400 studies are in the process or done in our network. For example, here's the, I'm sorry, it is written in Korean, Gyeongbuk National University Hospital. We can see the basic aspect of the databases. There are some databases in the hospital. For example, for the condition. Condition means the diagnosis. You see, what is this? Okay. This is the gastroesophageal reflux. Okay. From 2011 and 2017 for the age of 10 to 19, and for the 20s, for the 30s, 40s, like this, it changes. And for other hospitals, okay, this is the Gangdong Gyeonggi University Hospital. For the drop, okay. For the ticagrelor, the antiplatal agent. I think the drug introduced 2013 in that hospitals, and for the ages more than 50s, it is used widely, and the prevalence increased. The use of that drugs increased a lot recently. For the specific study, we need to use the atlas. For example, to select a specific cohort for a study, we use the cohort definition. I will click anything that was done in the previous one. I think they try to define anticoagulant used for the female, and we can define the definition using this GY, like this. Then it generates, and we can see how many patients we have by the criteria in that databases. In the characterization tabs, we can see the basic characteristics of the cohort we selected. In the code pathway, we can see how the patients was treated with the medicines. The inner circle means the post-treatment, the second circle means the second treatment, and third treatment. And most of the patients, they receive enoxaparin for their first treatment, and half of them receive this aspirin, and then another drug like that. And very small portion of the patients received rivaroxaban at their first medication, and then another drug and another drug like this. So we can see how the patients are treated. And for the incidence rate, also we can calculate incidence rate of a specific disease condition. Here, estimation, we can calculate the odds ratio risk of a specific medicine or disease for specific outcomes. This is the status of Korean Arts Network. As you see, now more than 60, the largest hospitals in Korea are joined to our network. Focusing on the COVID-19 study-thon, in this March of this year, we are planned to open the European Odyssey Symposium. However, as you know, due to the COVID-19, the meeting could not be held. Thus, the European Odyssey tried to open the virtual study-thon, online study-thon. However, for the study-thon, we faced that we have no available COVID-19 impactee data. At that time, Daegu province was endemic. Thus, we had COVID-19 patient data. Thus, with the help of the Ministry of Health and Welfare and the help of the Ministry of Trade, Industry and Energy, we could convert the 2 university hospitals' EMR data into MAP-CDM. Also, data, the claim data of the HERA. HERA is the only mandatory organization who collecting all the healthcare claim data all over the country. For the 2 university hospitals, we converted their EMR data into MAP-CDM, A hospital, B hospital, and through the network, we provided this data to the global researchers. We do not provide the data itself, but the researchers can access this data through the Findability platform. So that means the researchers cannot see the raw data, but they can only run analytic code against to this CDM data and get the result from this network. This is some of the characteristics of the Korean COVID-19 impacted in Daegu province hospitals. Interesting is young female patients was highly prevalent at that area. And of the pre-existing disease, there were high prevalence in essential hypertension and chronic kidney disease and gastritis like that. For the number of prescribed drugs, patients aged more than 50 received more medicine and received more treatment compared to other age groups. And for the HERA data, HERA is the Health Insurance Review Agency of Korea. It's a governmental organization. They received all the claim data from all the hospitals. We made this website, and the researchers can access 2 databases. One is the CDM data format. Another one is the data format for SAS. And the researchers can choose between the 2 databases, and the researcher make their own analytic code. And if they upload the analytic code into this website, then we run that analytic code into these databases and return the resulting statistics and graphics like that to the researchers. Fortunately, before the opening of the study-thon, more than 37 databases participated in the study-thon, and of them, 8 databases had COVID-19 patient data. And this is the screen capture of the study-thon. More than 360 researchers from 30 countries had participated in the study-thon for 88 hours using the Microsoft Teams. This is the list of the COVID-19 Global Collaborative Research Project performed with our Korean databases. As you see, 4 of them use CDM and the rest of them use the SAS data format because the CDM is still unfamiliar with many of the researchers. These are some of the results are already published, and some of them under review, and some of them are under the study in the progress of the study. This is an example of the published study using the databases. Here we try to compare the clinical characteristics of the patient having the COVID-19. This is the summarizing graph showing the difference between the databases. this is the database from the Columbia University in the United States, the distribution of age. But the Korean data, the distribution is somewhat different. There are some more young age groups in Korean data. The gray column means the patient, the historical data from the patient who took the influenza. This is the Spain data and the United States data. This is the characteristics of the patient between the countries. CUMC from the United States, here from the South Korea, like that. And as you see, the basic characteristics of the patients are different between the countries. And this result was published in Nature Communications. This is a study on the risk of mortality in elderly coronavirus disease, 2019 patient with mental health disorder, using the whole Korean population who took the COVID-19. We try to make a prediction algorithm whether a patient will be admitted, should be admitted to the hospital, or should be admitted to the ICU, or the patient will die. So using the Atlas tool, we try to make a prediction model. This is a chart enabling calculation of the risk without calculator. This is the web page calculating the risk of hospitalization, ICU admission, and mortality using that algorithm. Is an example for the, for example, the patient who aged at 42 and male and has a history of diabetes. Then, okay, let's calculate the risk. Okay, the patient has 2.9% of hospitalization and 1.1% of fatality and 0.8% of ICU admission. And if he has a heart disease, then the risk will be increased. And if the patient has hyperlipidemia, then the risk will be slightly reduced. Here, the age, we found that age is the most important risk factors. If the patient has aged more than 80, then the risk will be increased a lot. So during the support for the COVID-19 study-a-thon, we had lessons like these. The first one, the COVID-19 will not be the only pandemic disease coming. We think another new infectious disease can come in the future anytime. So the tight international collaboration between the countries is necessary to overcome the future disasters. And also we need to change perceptions while we try to provide our data, our COVID-19 patient data to the global researchers. There are many concerns providing our data to the international researchers. But as we see, the international data sharing or the joint research are not against the national interest. By providing our data to the international community, we can receive more benefits from the result of the researchers. So collaborative effort to cope with the common threats are necessary and mandatory for the future threats. Another things we face is the timeliness of the data availability. Although we had the CDM data at that time, however, that data didn't contain the COVID-19 patient data because we updated the CDM data biannually. So we have to download the data, the recent COVID-19 patient data, and we have to convert them into MAP CDM. So it takes a lot of effort and time, and also lots of other administrative barriers. Although we had technologies for the real-time conversion, however, some hospital information system was old, so they could not support the real-time conversion. And also we need to improve IRB process for this kind of research using anonymized data. it had required a long time before we get IRB exemption for the public use of COVID-19 data. For example, of the Columbia University, they receive one umbrella IRB for the whole CDM data for 1 year. So if by getting one IRB for the whole CDM data for 1 year, the researchers who use the data do not need any more IRB exemption for further researches during the 1 year period. Like this, we need to adopt that kind of IRB process. Also, we need a linkage between the public and private data for more detailed data. In Korea, in KCDC, they have epidemiologic data like the symptom, COVID-19 confirmation, mortality like that. And here they have reimbursement claim data. And for the hospitals, they have some more detailed rep result. However, these data are separated and we could not link between them. And the hospital data didn't use widely because they had not enough baseline period in the hospital, because most of the COVID-19 patients visited the hospital first in their life, so we could not have the baseline data in the hospital. But by algorithm of Atlas, we need at least 1 year of baseline period data. So many of the patient data in the hospital could not be used for the researches. If we could link between the hospital and here and KCDC, then we could get the more sophisticated research result. I think you may have interested in learning more about the RDC and the tools like the Atlas. Fortunately, we have textbook. The name of the textbook is The Book of RDC. In this book, you can understand more about the CDM and the tools used for the distributed research network. And the price of the book is about $10. It's very cheap. And also, there are free online book here, so you can access the data freely. Thank you for your attention. Today, I introduced to you about the basic principles of a common data model and distributed research network, and also introduced you about the Korean Odyssey and how we contribute to the study-a-thon on COVID-19 and the lessons learned during the preparation of the COVID-19 data in CDM format and provide that data to the global network.
I think this topic will be very nicely matched by the last speaker, Magnus Eriksson, a business architect of Swedish Research Council. The title of his presentation is Effective Data Sharing within COVID-19 Research. Please welcome Dr. Eriksson.
Hello, my name is Magnus Eriksson, and I work at the Swedish Research Council at the Unit for Strategic Data Resources. And my topic will be a bit similar to the previous presentation, but maybe from another perspective. So I'm going to share with you a little bit information on how we work with effective data sharing within COVID-19 research. And specifically how we work with enabling that. So the Swedish Research Council have increased the activities to enable effective and secure data sharing in the ecosystem over the last years. So we have been working with this for a bit, but of course the need for speed when it comes to data sharing and effectiveness has increased during the COVID-19 pandemic. So there are a lot of discussions on how to improve the data sharing and access to the research data sources. And one output that has become quite influential in Europe over the last years is the FAIR principles. And that is a set of principles that I thought I would mention a little bit more about in order to lay the foundation for our work. So the FAIR principles, it stands for findable, accessible, reusable, and interoperable. And it's a set of principles that was first minted in the life science community in 2016. And these are principles to follow if you want to enable efficient reuse of the data that you are collecting or producing. So the findability principle, it contains a set of principles that is about how to make it easier to find your research data set. So it contains metadata, minimal metadata on what you need. It contains a section on identifiers, how to uniquely identify your data, and how you need to go about providing this in a searchable resource like a repository or on the internet. The accessible principle, it contains a set of sub-principles that is about how to make your data more accessible using open formats and enabling authentication and access control, and also on the need to have your metadata separate from your data if you delete your data, so you can still have the metadata that describes how has this data been produced, so you can still have that knowledge. And that is also quite useful if you work with sensitive data, because then you might not be able to access the actual data, but you need to access the metadata separately before accessing this more sensitive data. And the final principle is reusability. And here the principles, or some principles, is about detailed metadata to describe the provenance, the context in how has this data been collected, how has it been processed, by whom, and so on. And this is done in order to for the researcher to be able to evaluate the data set or the digital object, and to see what kind of trust you can have in the data, and is it suitable for this study, and so on. In this principle, there is also a sub-principle that contains details about licensing, because that is an area that is sometimes forgotten. That it is very valuable to have a clear license on your data. And the interoperability principle, I think I forgot that one. It is about interpretation of meaning of your data. So it's a way to provide clear definitions about the content of the data. And we use ontologies and terminologies and such when doing this. And I heard in the previous presentation, I think you mentioned Snowman CT, and that is one of the terminologies that we use also. Okay, so these are the FAIR principles. One thing to think about when it comes to FAIR data is that it's a bit of a journey toward the FAIR data resources, because it's a lot of work that goes into to making your data FAIR, and it's important then to also support progress, because it's the most important thing is to have progress towards more FAIR data resources. Another thing is that it's very hard to achieve this by yourself. It's much easier to achieve it within an ecosystem, because you will be needing, as an example, service providers providing identifier services, or maybe semantic artifact services where you can link to terminologies or ontologies and such. So that is much easier. And finally, I think one thing to think about also is that it's very different when it comes to different scientific domains. Different domains have different starting points. So some scientific domains are very advanced when it comes to interoperability, as an example, and some are very advanced when it comes to findability. So there might be a need for different scientific domains to start working at different starting points when it comes to the different principles. And it's also, since it's a lot of work involved with this, it's also important to do a cost-benefit analysis, so you can see where do you get the most benefit from your resources. Okay, so at the Swedish Research Council, we have gotten 3 government commissions that relates to fair data. So the first one was a couple of years ago, and that one we have delivered. It was a government commission on to establish fair criteria to measure the involvement of the open science towards an open science ecosystem. And that was kind of the starting point, although the one mostly to the right, it was also quite early that we started to work with that. And that was a government commission to enhance the efficiency of data access when it comes to register-based research. And that one we are still working on, and it's still ongoing. And the one in the middle there, government commission on open access, that is also ongoing, and it concerns open access to research data. Common among these is that they are resting upon the fair data and the fair data management practices, because that is needed to enable these government commissions. And the fair data work that we are doing is done within different projects and initiatives. We are working within the European Open Science Cloud. That is a federation of services that is developed in Europe in order to enhance the reusability of research data. We are also working, we have created a national metadata repository where we have been working describing register data resources, both when with detailed metadata and also with semantic mappings to standard terminologies and ontologies. A SciLife lab that is a partner of us has developed a national COVID-19 data portal, and we have we are working with the coordination towards the European infrastructure, EMBL, EBI there. So that connects the different nations' COVID-19 data portals. And finally, I would also like to mention that we are working a lot with enabling support services, because there is a lot of training and the informatics work and so on that is needed to enable this. This is a short summary of the things that we are working on. As you see here, the EOSC web page here, you can see that the COVID-19 data portal comes up there too, because EOSC is a federation of services, and those services is often provided by infrastructure service providers such as EMBL, EBI, and that will then the SciLife Labs National COVID-19 Data Portal is one node in that hub. At the National COVID-19 Data Portal, we have also provided access to the National Health Metadata Repository, where we provide metadata on different registers, but in this case, maybe the most interesting ones are clinical quality registers of different types, and also biobank metadata, sample collections, and possibly also socioeconomic registers might be interesting here. And many things, many registers are in Swedish, but since we are doing the mapping to international terminologies, there is a possibility to go through that linkage to look at the resources. Okay, so and finally, the enabling fair support services. We work, all these technical things are very important, but even more important is to enable this with policy work, informatics work, metadata management, and semantic annotation and mappings. And I think I would guess that the previous the presentation in the previous with the previous presenter, that they are working with similar within similar areas. And the COVID-19 data portal, their SciLife lab is also offering similar services with bioinformatics, data management, and systems development. Yes, and of course, it's very important to start working with your FAIR data management in the beginning of the process, because if you are working with support services to enable this, even when you start with data management plans, data collections through data processing, data analysis, then when you are going to publish your data in a repository, then you already have access to your metadata and to your semantic annotations and so on that is needed to publish this in a fair manner. So then it's not that much extra work. It's much more work if you start looking at the reusability or reproducibility when doing the publication. Okay, so it's not only we that do this work. This is just a small subset of infrastructures that are working with this, that we are funding, and the European Spallation Source is working with it. Max4 is working with it. Elixir, it's a life science infrastructure, is doing a lot of work. And also within the linguistic area, the Clarin, the Common Language Resource and Technology Infrastructure. So it's an ongoing work, and it's very important to enhance the reusability of research data. Okay, so that is the end of my presentation.
And I'm afraid we are slightly behind the schedule, but we probably have 5 minutes of short discussion. And probably, I guess, Dr. Park and Dr. Erickson may share common interest on data sharing. So I would like to ask Dr. Park to initiate your discussion, please.
So I'm impressed to the project you're leading, Dr. Magnus Eriksson. So I think the two projects are very similar in their basic principles, and I think we can learn more from each other's project. And I would like to ask whether you know about the Eden project, supported from the IMI2. Europe, I think more than 12 European countries are joined into Eden project, and they are trying to convert the hospitals EHR data into a common data model, and also they are educate small to medium business companies to let them train and let them convert the hospital EHR data into the MAPSIDEM, and let the researchers can use of them. I'm sorry.
Yes. Well, then I have heard of it, but we are not participating in it. But the work that we are doing is, of course, laying a foundation for the harmonization work that is needed when assembling or transferring data into the common data model. Yeah.
Okay.
What is your? It's very interesting to hear about your work with such a large implementation of the common data model. What do you think was the most challenging sections there when it comes to data management and semantics?
Yeah. I think the technical issue is not big deal. The big deal is motivating the hospitals. Usually, the hospitals do not want to share their data and to other researchers. So the most challenging part is the motivating and persuading the hospitals. And the others, the technical issues, already we have many tools because the Odyssey started in 2013, and we have many softwares, and all the software, the source codes are open to the community, and it's developing, updating daily. So I think, the data conversion and the quality insurance and the tools utilizing that software are already done enough?
My question is not directly related to any of the talks today, but overall, I'm curious and ask everyone, speaker in this session and previous sessions, about our preparedness for next pandemic. I mean, I guess there are many potential pathogens out there, and there's also WHO blueprint they have prioritized. But after this pandemic, what do you think? I mean, what kind of pathogens that we need to be prepared for? I mean, as a molecular biologist, I'm curious which topics that are needed to be studied by biologists and medical researchers. So could you answer my question? I think you're the expert.
Well, probably Dr. Kim knows better answer than myself because she is an RNA biologist and much knowledge, accruing much knowledge on the immune evasion strategy of the RNA viruses. As she mentioned, the WHO lists about 10 different pathogens. All of them are RNA viruses that would cause the pandemic in the next decade or two. Probably the best way to guard against such a pandemic, outbreak of pandemic, is to establish, from the vaccinologist's point of view, is to establish a rapid production platform of the vaccines, as opposed to the classical vaccine development platform that requires at least 10 or 15 years of development. So the establishing a common platform that would enable the development and the deployment of the novel vaccines as soon as possible, that would be the common interests of all the participants, not only in Korea, not only in Sweden, but everybody in the whole globe, to be better prepared for that. I'm not sure it may suffice your question.
Well, probably I think I can talk about just a little bit about in terms of drug development, because there are a lot of things in terms of diagnostics and the vaccines and then drug development. From the drug development, I think there was actually some alarm. I mean, even before this pandemic, like 5 years ago, there was some report from the coronavirus virologists that maybe the SARS-like coronavirus maybe just appear, and then we are somehow just expecting the occurrence of these SARS-like coronaviruses, and then if we just took this very seriously, at least we could just prepare just well before this pandemic occurs. In terms of the drug development, well, there are some prior good examples of successful drug development. For example, like HIV and HCV, and then mostly the protease inhibitors and then RDRP inhibitors were pretty successful. And then coronavirus also just belongs to the RNA viruses. So I think we can imagine that the development of some pan-coronavirus inhibitors targeting the proteases and the RNA-dependent RNA polymerase, and that should be one of our just long-term goals. And even after this the pandemic, that we prepare for another just a coronavirus pandemic.
I guess Dr. mentioning the pan-corona antiviral may be equivalent to the universal vaccine against corona. And so far, the vaccine is depending on the specific immune response that provides specific response to the particular antigen, particular virus. Probably the next step and the common homework or future challenge for us to develop is to provide a vaccine providing a cross protection against many genetic variants. It would be a real challenge because it challenges the core of the immunology. But we know that some vaccine provides is a very strong inducer of innate immune response, but innate immune response providing broad protection. The drawback of innate immune response is that the effect is very ephemeral. It only goes about a week or maximum 2 weeks. If we can combine, if we can delay somehow the transition from the innate immune response into a specific immune response, adaptive immune response, then this type of technology could be combined to develop a universal vaccine, or the vaccine they provide broad protections against genetic variants. I guess we are slightly behind the schedule. Probably I have to wrap up the COVID-19 session of the second Korea-Sweden R&D Network Day. And I thank all the participants from both countries and the opportunity to share and discuss recent findings on this novel virus. The forum today, I hope, will serve as a common ground for future collaboration between the two countries. But on the other hand, I honestly hope the COVID-19 is the last topic to discuss in this network, and hope to meet again next year on a brand new topic without any facial mask. Thank you for your participation.
Thank you.